Monohybrid cross, a basic genetic experiment. A monohybrid cross is a type of genetic cross where two individuals are crossed, each with different alleles for a single trait. This trait is controlled by a single gene. The goal of a monohybrid cross is to study how this trait is inherited from one generation to the next. Mendel's experiment. Gregor Mendel, a 19th-century Austrian monk, is often credited with discovering the basic laws of inheritance through his experiments with pea plants. One of his most famous experiments was a monohybrid cross, where he crossed pure-breeding pea plants with either purple or white flowers. Mendel's hypothesis. Each trait is controlled by a hereditary factor, now called a gene. Each gene has different forms, now called alleles. Each individual inherits one allele from each parent. The dominant allele will mask the expression of the recessive allele. Punnett squares. Punnett squares are a simple tool used to predict the possible offspring genotypes and phenotypes from a cross. Example: let's consider a monohybrid cross between two pea plants, one with purple flowers, PP, and the other with white flowers, PP. Genotypes: PP, PP, PP. Phenotypes: purple flowers, white flowers. Key terms: dominant allele, an allele that masks the expression of another allele. Recessive allele, an allele whose expression is masked by a dominant allele. Genotype, the genetic makeup of an individual, represented by the combination of alleles. Phenotype, the observable physical traits of an individual, determined by the genotype. Homozygous, having two identical alleles for a particular trait, e.g., PP or PP. Heterozygous, having two different alleles for a particular trait, e.g., PP. By analyzing the results of a monohybrid cross, Mendel was able to deduce the laws of segregation and independent assortment, which form the foundation of modern genetics.